All right, so today we are going to go through a list of the organelles that are found principally in animal cells. So next week we are going to focus on those organelles that are specific to plant cells. All right, to get started, something that is found in both plant and animal cells is the nucleus. And in general, the role of the nucleus in the cell is to control all cellular activities. It's also where the genetic information is stored in the cell. Within the nucleus, there are a few different components that you'll need to be able to recognize. One is the nucleolus. So that almost looks like it's the nucleus of the nucleus. But this piece is used to make ribosomes. We'll talk about those in a few slides. The nuclear envelope is the cell membrane of the nucleus. And it's actually made of two layers of membrane, two phospholipid bilayers. So imagine the bilayers that you see in a normal cell membrane. You have the heads and tails facing inward towards each other. The nuclear envelope is made of two layers of those phospholipids. But in the nuclear envelope, there are things called nuclear pores. So these are channels that do allow things like RNA and other materials to pass in and out of the nuclear envelope. Also within the nucleus, similar to a cell in general, there is a nucleoplasm. So that's like the cytoplasm of the nucleus. Again, a jelly-like liquid that helps hold things in place. Moving on to our mitochondria. So the mitochondria are the powerhouse of the cell, and their main job is to create ATP from glucose. So the more active a cell is, the more mitochondria it has, because it needs to produce more energy in order to function. So as an example, muscle cells can have hundreds of mitochondria, whereas other cells that aren't involved with as much activity and movement would have fewer mitochondria. The mitochondria also has two layers of membrane. It has kind of a smooth outer layer and then this kind of curly, swerved inner folds that make the second level of the membrane. And these are called cristae. So these, again, are the inner membrane layer that make those kind of folds. So within the cristae, this is where different chemical reactions are taking place in order to help generate energy. As you can see here in the center, mitochondria also have their own DNA. So mitochondria are interesting because they are the organelles that only reproduce by dividing their individual mitochondria. So mitochondria come from existing mitochondria. They're not created by other organelles in the cell. So because they need to divide and split to make more mitochondria, they have their own DNA located within the organelle so that it's ready to go when they need to divide. Taking a look at the energy created in mitochondria, just as a review, I believe we went over this reaction a few lessons back, but ATP, adenosine triphosphate, is the energy currency in cells. In other words, ATP is the energy needed for all cellular activities to take place. And within the mitochondria, that is where this ATP is generated. So the mitochondria take in glucose and oxygen, and as waste products, they expel carbon dioxide, water, and then again, this ATP that the cell can then use to do anything it needs to do, divide, grow, creating other organelles or other molecules for life. Moving on to our ribosomes. So ribosomes are organelles that are in charge of making proteins in cells. So they are these round little organelles that have no membrane, and they're just made of protein and RNA. Remember, RNA is what the body uses in order to code and create proteins. So ribosomes are created starting in the nucleolus. Remember, that's like the nucleus of the nucleus where ribosomes and proteins begin. And then the process is finished in the cytoplasm. And some are floating around in the cytoplasm, but others are attached to the endoplasmic reticulum, specifically the rough endoplasmic reticulum. Speaking of which, the endoplasmic reticulum. So that endoplasmic reticulum, or ER for a nice abbreviation, is considered the intracellular highway. So it's the area where different molecules are created and transported within a cell. And there's two components to the ER. We have the rough ER and the smooth ER. The rough ER, as you can see here, is covered with ribosomes. And the role of the rough ER is to make phospholipids and proteins. The smooth ER, which you can see over here, again, smooth because it has no ribosomes stuck to the outside. That is where lipids are created, for example, cholesterol, estrogen, testosterone, and other hormones. Moving on to the Golgi apparatus. So the Golgi apparatus is used to package molecules to get them ready for leaving the cell or being exported out of the cell. So the Golgi apparatus kind of looks like the endoplasmic reticulum, the rough endoplasmic reticulum. It's made of these folds and curves and swerves, but the goal of the Golgi apparatus is to take molecules that are produced in the cell and modify them and essentially get them ready to leave the cell. So if those molecules need to be modified in any way or labeled for what they need to be used for, that's what the Golgi apparatus does. So it takes all those molecules that are ready to leave the cell and make sure that they are prepared to go and in the proper form. Lysosomes. These are organelles that help to break down things that the cell doesn't want or no longer needs. So breaking down things like bacteria, large molecules, old organelles that aren't really working the way they should anymore, and the cell needs to get rid of them. So lysosomes are vesicles that are budding from the Golgi apparatus. And vesicles have a lot of different roles in the cell, but lysosomes are a specific type of vesicle that, again, breaks down things that the cell doesn't want or doesn't need anymore. Inside the lysosome, it has digestive enzymes. So remember, enzymes are molecules that help chemical reactions to take place at a faster rate. So it helps to speed up digestion of things that the cell needs to get rid of. Also, another word to note is autophagy. So that's when a lysosome breaks down an old organelle that doesn't need to be used anymore. So the lysosomes are kind of like our cleanup crew. Moving on to the cytoskeleton. So the cytoskeleton in this picture is all of these like yellow fibers that you can see there. These are protein fibers that crisscross the cell and help to support the cell. So it's made of thin tubes and filaments. Filaments are kind of like stringy things, but what they do is they act like tent poles in a tent. So imagine a tent without tent poles, it would just fall flat and not stand up. So the cytoskeleton are like the tent poles in a cell that help the cell to keep its shape, so it doesn't just collapse and squish in and on itself. Centrioles. So centrioles are only found in animal cells. Very important to note. And centrioles are these guys here. They're special organelles that are only active during cell division. So if you think back to past years when you learned about the steps of mitosis and meiosis, you might have remembered seeing these guys, these centrioles, that attach fibers to chromosomes and help split them in half, so that half the genetic material goes to one side of the cell, half goes to the other, and then the cell can split into two. So the centrioles are what have these spindle fibers that attach to the chromosomes and pull them apart. And again, they're only active when the cell is dividing. If the cell isn't dividing, they're just hanging out. They don't really need to do anything. Last here we have cilia and flagella. So these are really just relevant for our single-celled organisms, like archaea, bacteria, protists. In the gifts that you see here, these are both protists. We have our paramecium here, and we have our euglena down here. So cilia and flagella are hair-like structures used for movement. And as you can see up here, in our paramecium, there's like these little hairs all around the outside of the paramecium that kind of flip back and forth like fins. So those are cilia. And our flagella are these long whip-like tails coming from our euglena.